Okay. You can see it now. So, Campbell 17 is gene expression from gene to protein. So this is basically how your proteins get formed. Okay. So the first thing is the central dogma. So as we've talked about before, there are differences between RNA and DNA. So DNA is single-stranded, and then DNA is double-stranded. RNA is single-stranded. RNA contains uracil instead of thymine, and it also contains ribose instead of deoxyribose as its 5-carbon sugar. So the central dogma is basically the process of converting. Hold on, give me a second. Okay, sorry. It's the process of converting the genetic code into proteins, which has 2 separate processes. So the first process is transcription, which is where the RNA is synthesized using the information in DNA. So all the DNA information is transferred into RNA. So that's why it's called that's why it's called transcription, because you basically transcribe the information. So the first type of RNA we will be talking about and we'll see is messenger RNA or mRNA. So that carries the information into ribosomes. So the next process is translation, where the polypeptides are synthesized using information carried in mRNA. So the mRNA, you can think of that as like basically being translated into another language, which is amino acids. So you go from nucleotides to amino acids. What? Are you saying something? No, okay.
What I feel like I've heard the word dogma before, though.
You probably have, maybe. Well, that's what it's called, so you'll be hearing that a lot. Okay, so both transcription and translation will occur in both prokaryotes and eukaryotes, but the locations are a little bit different for the two. So in eukaryotes, transcription will occur in the nucleus, and then translation will occur in the cytoplasm, like like outside of the nucleus when the mRNA is brought out of the cell. But remember that prokaryotes don't have a nucleus. They have a nuclear region, so the transcription and translation will basically occur in the same space simultaneously. So I'll talk a little bit more about this later, but basically when the cell is doing the transcribing, the new RNA is also getting translated at the same time. Okay, so this is just the difference between prokaryotes and eukaryotes. So in prokaryotes, they don't have a separate nucleus. They don't have a separate nucleus space. So transcription and translation will both occur in the cytoplasm. But in eukaryotes, they have a nuclear envelope. So when your pre-mRNA is processed into mRNA, that will go out of the nucleus and then be translated in the cytoplasm. Okay. So next is the genetic code and how that transcribes into RNA. So in DNA, there's only 4 nucleotides to express more than 20 types of amino acids. So this means that each protein can't have its own nucleotide. So then the cell solution to this is to basically have amino acids coded by triplets or like groups of 3 nucleotides for 1 amino acid. So then this allows for a lot more arrangements of nucleotides for each amino acid, since you kind of have different combinations of nucleotides. So during transcription, only one of the DNA strands is used for replication. This strand is called the template strand. So therefore, like the synthesized RNA, the RNA that you make from the template strand is complementary to the template strand, since we know there's base pairing going on during transcription. So since this synthesized strand is complementary and it's going to have an identical sequence to the non-template strand, which is called the coding strand, since that information will be passed on into the RNA. So then after transcription, the mRNA triplets are called the codon, which is usually written the 5' to 3' direction, and that will basically encode for your protein. Okay. So the next thing we're going to be talking about is codons. So the codons in the mRNA are They basically dictate what types of amino acids will be translated. So like I've said, each codon of 3 mRNA nucleotides will code for one amino acid. So something good to know is AUG. So AUG will code for methionine and it's also called the start codon. So whenever methionine is coded for, the protein will start to form. So also during translation, the reading frame or like where the codons will be so how the codons are read are very important, or else all of the following proteins can be coded wrong, and then the polypeptide can have serious issues with that. Okay. So this is the actual process of transcription. The main enzyme during transcription is called RNA polymerase. So it's kind of like during DNA replication where the main enzyme is called DNA polymerase. So this polymerase will just add on nucleotides for RNA. So here RNA polymerase will first split the two DNA strands apart. Since the strands are connected by hydrogen bonding, you can split that apart. And then it will add complementary RNA nucleotides to the growing RNA strand. So this is a little bit different from DNA replication because this process doesn't need a primer to begin adding RNA nucleotides, but the polymerase has to attach at a promoter region on the DNA and then detach at a terminator region on the DNA. So a really common and important promoter is called the TATA box, the TATA box, which is a sequence that has repeats of thymine and adenine. So the direction of transcription is usually referred to as downstream, while the other way around is referred to as upstream. So specifically, prokaryotes will have a single type of RNA polymerase, and eukaryotes different types of RNA. So the specific polymerase that synthesizes pre-mRNA, so pre-messenger RNA, is called RNA polymerase 2. There's something else, the portion of DNA that's transcribed is called the transcription unit. And so in transcription itself, there's 3 different parts. So the first part is initiation, the second part is elongation, and the third part is termination. So during initiation, the RNA polymerase will unwind the DNA and then attach to the template strand at the promoter to initiate transcription. So there can also be certain transcription factors that are proteins that will bind to the DNA and help the RNA polymerase attach. This transcription factor and the RNA polymerase together will form the transcription initiation complex. During the second step, which is an elongation, RNA polymerase will add complementary RNA nucleotides downstream and then elongate the transcript 5' to 3'. When the RNA polymerase moves along the DNA will rewind itself because the nucleotides can repair with hydrogen bonding. So lastly, at termination, the RNA polymerase and pre-mRNA are released, and then the DNA will rewind itself. So this is done when RNA polymerase transcribes the polyadenylation sequence, and then that basically causes the RNA polymerase to detach from the protein. So here you can see the 3 steps. So first is initiation, where the RNA polymerase will bind, elongation, where the RNA is made, and then termination, when everything's released. Oh, sorry. Okay. Next, the initial product of transcription is called pre-mRNA, or the primary transcript, and this primary transcript has to be processed to become usable mRNA. So during alteration, a 5' guanine cap and a 3' poly A tail will be added to the 5' and 3' ends. So these caps and tails will help with the export of mRNA and also protect against mRNA degradation. Since mRNA is single-stranded and it can get degraded easily. There will also be untranslated regions in the mRNA that are not translated into proteins. So another process in RNA modification is called RNA splicing. So during RNA splicing, a lot of RNA will be removed and then the remaining sections are reattached. So during RNA splicing, enzymes called spliceosomes will help remove introns, which are the non-coding portions of the mRNA. and then splice together exons, which are the coding portions. So if you want to remember exons and introns better, it's good to remember that exons are expressed while introns are not. So they're the intervening sequences. So RNAs can also splice itself and act as enzymes, so they actually help with the spliceosome action. So this is because RNA is single-stranded, and then so its base pairs can pair with itself to basically form a specific three-dimensional structure. So there's also certain base pairs in RNA that have functional groups that can help with catalysis. So these RNA base pairs can pair with itself and also form hydrogen bonds to other nucleic molecules so that they can perform enzymatic activity. So in spliceosomes, there are small RNAs that act as ribozymes to help cut out the intron sections. So RNA splicing is very useful for creating variation in individuals and expression. So sometimes certain exons are spliced out as introns, and this creates more variation in the mRNA that is shipped out of the nucleus because in certain mRNA you have, like, for example, a certain exon, but then in other mRNA that is transcribed from the same DNA, you don't have that exon, so the proteins will be a little bit different, and that creates a little more variation. Okay. Next, during translation, a transfer RNA or a tRNA will bring an amino acid to the corresponding codon, the corresponding codon in the ribosome. So each tRNA is made of a single strand of RNA that will fold on itself because of the hydrogen bonding. And on the bottom, it will contain anticodons that pair with codons in the mRNA. So each tRNA has its own amino acid attachment site where the amino acid will be added through. So on the top here. And then on the bottom are the anticodons. So the tRNA is matched with its amino acid by amino acetyl tRNA synthesis. And there's about 20 different syntheses, one for each amino acid. So that's good to know. There is also a phenomenon during translation called wobble. So during wobble, slightly different codons can still code for the same amino acid, and this helps mitigate the effects of mutation of single nucleotide mutations. So in wobble, the third nucleotide base of a codon can slightly vary, but then this still will result in the same protein. So in the same amino acid, sorry. So here, as you can see, for example, for leucine, you can have the last nucleotide can be slightly different, or the first one can be slightly different, but they will all code for leucine. So if there is a mutation in the codon that slightly changes the last nucleotide, it will still code for the same amino acid, so that prevents like excessive changes in the amino acid when there is a change. Austin, are you falling asleep? Okay, it's okay.
What the hell?
Actually, pay attention, please. Okay. Okay. Translation is carried out in ribosomes. So ribosomes are made of ribosomal RNA or rRNA that is put together in the nucleolus, and also some proteins that will hold the structure together. So ribosomes are composed of a small and a large subunit that will attach to the mRNA. So the ribosome contains specialized sites for RNA synthase, which are the A, P, and E sites. So the A site holds the next tRNA that needs to be added to the amino acid chain, and it's also it's called the amino acetyl tRNA binding site. So that's the tRNA and amino acid complex that we just talked about before. So next, the P site will hold the growing polypeptide chain as the amino acids are transferred to the growing chain. And then lastly, is the exit site, so E for exit, which the used tRNA will exit the ribosome. So the used tRNAs are not degraded. They can still have new amino acids added onto them. Okay. During translation, the code AUG, which codes for methionine, will signal the initiation of translation. So after GTP is spent for energy, the large subunit and then the small subunit will attach and start translation. So the first step in translation is codon recognition. So in codon recognition, these tRNAs with their amino acids will try to match with the codon. So whichever one matches first will be attached to the codon and then move into the P site. There are also going to be a lot of other tRNA with different anticodons, but only the one with the matching anticodon will be able to bind. So the next step is peptide bond formation. So here the ribosome will act kind of like a catalyst and help with the formation of the peptide bond between the amino groups of the first amino acid and then the next amino acid. So the last step is translocation. So here the tRNA will be moved back a site. So the one in the A site will be moved to the P site. The one in P site will be released through the E site. So the mRNA will basically move along with the tRNAs and then can bring the next codon that needs to be translated into the A site. Because here's just a diagram of everything I was talking about. So first is codon recognition, where the tRNA comes into the A site. Next is peptide bond formation, where the peptide bond will form between the amino acids. And then third is translocation. So then the tRNAs will move back and then leave a new A site open for the next tRNA to come in. Okay, translation will stop once the stop codon is reached in the mRNA. So when the stop codon is reached, the ribosome will basically take in a release factor instead of another tRNA. So it's a little bit similar to a tRNA, but it won't have any amino acids on it. So the ribosome will take one of these and then release itself from the mRNA. So after translation ends, there can be other post-translational protein modifications carried out. And these include adding sugars, lipids, phosphate groups, and other functional groups. So this is how glycoproteins can be made, since sugars can be added to the proteins. Okay. But the proteins will have to go somewhere after they are released, right? So how do they know where to go? So essentially, free ribosomes will synthesize proteins that stay for intracellular use, since the protein is directly released into the cytoplasm. The other type of ribosome is bound ribosomes that are stuck onto the rough ER. So these are secreted through membranes or out of the cell. So all protein synthesis will begin in free ribosomes, but the signal peptides, so here are signal peptides, they will cue the ribosomes to bind to the rough ER. So this signal peptide on the newly formed polypeptide will bind to a signal recognition particle, or an SRP here, which will then attach to a translocation complex on the ER. So then the translation will continue, and then the polypeptide is synthesized directly into the ER and then dropped off, and then it can be transported. Okay. So here's a little bit about polyribosomes. So in both eukaryotes and prokaryotes, there can be many ribosomes that translate an mRNA strand simultaneously to quickly make polypeptide copies. So these ribosomes are called polyribosomes or polysomes. And actually prokaryotes have to use this method since they don't have membrane-bound organelles. Actually, no, that is not related, but so there's basically just many ribosomes synthesizing the same protein out of the same mRNA. Okay, so here's the big diagram of everything that happens. So first, just a refresher, is transcription, where RNA is transcribed from a DNA template and all of the DNA information is moved into the RNA. Second is RNA processing, where the pre-mRNA is processed into mRNA. So that involves adding the poly A, the 5' guanine cap and the poly A tail, and also splicing out introns and leaving in exons. But during that process, during that process, alternative splicing can also occur to allow for more variation. So then the tRNA will be attached to an amino acid by the aminoacyl tRNA synthase. And then next is translation, where the tRNA will bring their amino acids into a ribosome and then allow the polypeptide chain to grow. And then after the polypeptide chain finishes growing, it can also undergo post-translational modifications to add other groups to it. Okay, so here's some mutations that can affect the polypeptide chain. So most mutations are point mutations, which are changes in single nucleotide pairs. So the first type is substitution, which is the replacement of a nucleotide and its partner with another nucleotide and its partner. So for example, if you have two cytosines and a guanine, it can be substituted into two cytosines and an adenine. So these can be divided into like three groups. So the first type is a silent mutation. So the nucleotide chain will not actually change the resulting amino acid because of wobble. So like I've said before, so many so the last one of the three triplets can usually be altered for the same amino acid to be produced. So the next is missense mutation, where the nucleotide chain will result in a different amino acid being produced. And the third type is nonsense mutations, where the nucleotide chain will result in an early stop codon. So all of the amino acids after that will not be produced. So next is insertions and deletions. So these are losses and gains of nucleotide pairs in a gene. So these can be frame shift mutations. So that will shift the entire reading frame of the nucleotide. So the amino acids that follow will change drastically because everything is changed. So for example, here, if you have the red dog and you delete the T, the reading frame will still be, so this sequence will still be read in groups of three nucleotides. So everything will be changed and all of the amino acids produced from it will be different. So mutagens can actually interact with DNA in ways that can cause mutations. So for example, like X-rays and UV rays, this is also why you should wear sunscreen. And mutagens that cause cancer are called carcinogens. Okay. Okay, I'm gonna stop share. Do you have questions? Hot tub box? Yes, it is thymine and adenine. Okay, wobble is like where it's like a phenomenon where you can have, so you know how there are three nucleotides and a codon, so it's usually the last nucleotide can also be changed slightly, but the same amino acid will still be produced. Yeah, CCG is just the codon I was using as an example. Okay. Okay, next is Campbell 18, which is regulation of gene expression. Okay, so it's important that not all the DNA is being transcribed all the time to prevent like production of unnecessary things when they aren't needed. So the cell will have to regulate transcription so that it doesn't happen all the time to all of the genes. So bacterial cells, they use operons that are sections of DNA with different components that will transcribe into different enzymes to help with regulation of transcription or production of substances only when they're needed. So a very common example of an operon is the lactose or lac operon, which produces enzymes for lactose digestion only when there's lactose in the cell. The operon has a lot of different parts. So the part most upstream, so like the most before the coding portion is called the regulator gene, which will transcribe into the repressor. So right before the operator and the coding gene, the promoter will basically allow for the RNA polymerase to attach. So the repressor, which was initially produced in the regulator gene, will bind to the operator, which is right before the coding gene. So when the repressor is bound, it will basically block the polymerase from transcribing the gene. So you can just kind of think of the repressor as, so here's your polymerase, here's your repressor. So it will not be able to pass because of the repressor blocking its way. So operons can participate in the name of the book. It's called hold on. Okay, so this is the one I have. It's called Campbell by Biology. Yeah. And it's this thick. So oh, and the one I have is the 11th edition. Yeah, okay. Anyways, so operons can participate in two main types of gene regulation, which are inducible and repressible. So inducible operons are always off unless they're turned on by the target molecule. And on the other hand, repressible operons are always on unless they're turned off by the target molecule. So the lac operon that we've mentioned before is an example of an inducible operon. So when lactose accumulates in the cell without the primary source of energy present, which is glucose, the repressor will basically will then leave the operator and bind to free lactose that allows for the transcription of the lac gene to create enzymes that will metabolize lactose. So the other main type, or actually not main, but like very well-known type of operon is the tryptophan, or it's called the trip operon. So that's an example of a repressible operon. So the tryptophan will act as a co-repressor. So basically, when there's excess tryptophan being produced by the gene, it will accumulate and bind to the inactive repressor to activate it, which then will bind to the operator to stop tryptophan production. So that's how it's always on, but it will be turned off by the target molecule. So something else. So cyclic AMP will also help with positive gene regulation. So when glucose levels are low, cyclic AMP will then accumulate, which then binds to another CRP protein, which then binds to a specific site upstream of the lac promoter. So this will actually act kind of like a transcription factor in a way, in that it will help with RNA polymerase binding, which then can produce more enzymes to metabolize lactose. So basically to replace the main energy source, which is glucose. Okay. So here is a diagram of the lac operon. So usually the lac repressor is active. So usually it's bound on the operator here. But then if you have a lot of lactose, it will bind to the repressor, basically inactivate it, and then the RNA polymerase can go and transcribe the lactose enzyme genes. So here is the tryptophan operon, the trip operon. So usually the repressor is inactive, but then when there is too much tryptophan, that tryptophan will bind to the repressor, activate it, and then block transcription of genes for tryptophan. Okay. So all cells will have of an organism will have the same genome, but different types of cells will have different phenotypes and basically what they look like and characteristics. So they will basically express different parts of that genome. And this is called differential gene expression. So this expression isn't controlled by having parts of the genome physically cut out, but it's actually due to regulation of chromatin structure. So the first type is called histone acetylation. So this is when acetyl groups are added to the histone proteins in chromatin, and then this will loosen up the chromatin basically, and will help and promote transcription. So the other type of differential gene expression is methylation. So DNA methylation will cause the DNA to be more tightly packed into heterochromatin. Wait, did I mess? Okay. So acetylation will cause the DNA to be more loose. Methylation will cause the DNA to be more tightly packed. So methylation will make DNA less accessible for transcription. So this methylated DNA usually will stay methylated and be passed to daughter cells, and this is called genomic imprinting. So this is why, for example, liver cells will only divide into liver cells, and they won't divide into cells that are expressing everything. And so these are types of epigenetic inheritance where changes in expression don't depend on changes to the nucleotides. They actually depend on changes in chromatin structure, and these can technically be reversed. Okay, so in general, transcription factors are needed for transcription of all protein coding genes. And the transcription complex has to be assembled first for RNA polymerase to attach. So these transcription factors will basically help the RNA polymerase stick to the promoter. However, there are some genes that are not as heavily expressed, and that requires specific transcription factors. There are also enhancers, which are very distant up or downstream of the gene that can help regulate the gene. And there are also combinations of control elements that can regulate transcription. So all of these types regulations of transcription are not due to changes in the DNA itself, but it's due to like outside factors. Okay. So there are also post-transcriptional regulations. So alternative splicing, as we have mentioned before, involves splicing out exons as introns, and so this creates more variation in the genome. It also can control what is expressed. mRNA also, since it's single-stranded, it will be degraded in eukaryotes within several hours or days, so that prevents excessive translation of the mRNA, and also limits the amount of product that can be produced. And in plants, translation can be triggered by external factors such as light. So I guess that's good to know as well. Okay, so non-coding RNAs will also influence gene expression and translation. So microRNAs are the first type. These are essentially small, short strands of RNA that will bind to a complementary mRNA. So when it does this, it will block transcription of mRNA or initiate its degradation. So this is a microRNA here. So if the microRNA is completely complementary to the target mRNA, the mRNA will be degraded. But if it's only partially complementary, the mRNA will not be translated. So small interfering RNAs are kind of similar to microRNAs since they also bind to complementary mRNAs. But it is a little bit different since the small interfering RNAs will regulate gene expression, while the microRNAs will inhibit or degrade the mRNA. So next, Pee-wee interacting mRNAs will induce the formation of heterochromatin to express the expression of DNA, to slow the expression of DNA. So heterochromatin is the tightly bound form of DNA. There are two types of chromatin, which I've said, hetero and euchromatin. So euchromatin is loosely bound and can be easily expressed, while heterochromatin is densely packed and not accessible for transcription. So lastly, long non-coding RNAs are responsible for X chromosome inactivation. So in some cells of a female organism, one of the X chromosomes will be inactivated randomly. So these long non-coding RNAs will basically bend back and coat the entire heterochromatin and cover the genetic material and prevent transcription of the DNA in that X chromosome. So X chromosome inactivation is actually quite common. So for example, in calico cats, so female cats with like 3 different colors, white, black, and orange patches, they have X chromosome inactivation. So this can also occur in humans. So I think for humans, some patches of skin will not have sweat glands or something. I'm not quite sure, but you guys can read up on that if you're interested. Okay. So when multicellular organisms develop, their cells are going to differentiate and become different types of cells that will specialize in different functions. So the process that will physically differentiate the cells into different shapes and physical characteristics is called morphogenesis. So this process can be traced back to the changes in shape or other characteristics of the cell in different parts of the embryo and what they will be used for later. as the organism grows. Okay, so how do cells know what types of cells to divide into after the zygote is fertilized? So first, the egg cytoplasm that comes from the mother will contain a lot of cytoplasmic determinants. So these determinants will help decide which part of the zygote will differentiate into what type of cell. So after the egg is fertilized, the zygote will divide depending on the concentration and spread of these cytoplasmic determinants. And then the new daughter cells will divide depending on what determinants it has from the parent cells. Also, induction between embryonic cells can also determine the way cells behave and divide. So signaling molecules can be passed between embryonic cells and cause changes in the target cells. So these cells can like tell other cells, hey, like you need to divide into X, like Y type of cell, and then they will divide into like X type of cell. Okay. So after cells undergo determination, they will form the committed cell, basically. So these determined cells are going to express tissue-specific proteins from master regulatory genes and other tissue-specific genes that make them different from the other types of cells. So, for example, liver cells will express the albumin protein, while lens cells will express crystalline protein. So these proteins make them different from each other. It's good to know though that the tissue-specific genes have to be present for the master regulatory genes to also work during determination. So the cells have to actually be committed to being that specific tissue for these proteins to be produced. Okay, so different processes can affect cell determination, but the most common is transcription and translation, since that allows for tissue-specific proteins to be expressed. Oh, okay. Okay, so these determinants, the cytoplasmic determinants that we've talked about, will help determine pattern formation, which is the decision of where the organism's axes are going to be before organ development. So the cytoplasmic determinants will provide the positional information and will tell where a particular cell is compared to the other cells around it. So pattern information is controlled by these determinants, but it's also controlled by homeotic genes, which basically determine what develops where. So in the top right here, the fly had a mutation. Sorry. I'm going the wrong way. Okay. So in this fly, in this fly it had a mutation. And so one of the antenna was developed as a leg, while this is the normal wild type. Okay, so these homeotic genes will be provided from the mother because the mother will donate its like cytoplasmic determinants and also its a lot of the cell itself. Okay, so these homeotic genes will also be called egg polarity genes because they help determine the polarity of the egg and where the axes are. How can it carry so much weight with tiny legs? I don't think these legs are actually for carrying weight. It's just a mutation, so I don't think they're gonna use them, actually. They might. I'm not sure. Okay. Okay, so these homeotic genes are called egg polarity genes because they will help determine the axes and polarity of the egg. So one group of these genes will set up the anterior and posterior orientations of the egg, while the other group will set up the dorsal and ventral axes orientations. So these mutations in the homeotic genes of the mother or the maternal effect genes that will determine the cytoplasmic determinants will result in faulty development of offspring regardless of genotype. So since these are passed on from the mother, if the mother has a mutation in the cytoplasmic determinant genes, the offspring will obviously have those as well. So these mutations are often embryonic lethals, which means they will result in the death of the embryo. So an example of one mutation in the homeotic gene is the bicoid gene. So the mother has two mutant bicoid alleles that have that will basically produce embryos that have two like butt ends, so two behind ends. So bicoid is usually concentrated at the anterior end and determines the rear end of the embryo. And this bicoid protein actually contributed to the morphogen gradient hypothesis that basically says that morphogens are substances that will determine the local morphology of an embryo or like what it develops into. So as you can see here, a mutation led to two tail ends of the embryo where it should be a head and a tail end. So this embryo obviously can't survive since it doesn't have a head, so that's called an embryonic lethal mutation. Okay, so as I've just said, morphogens will establish the embryo's axes and other features. And the morphogen gradient hypothesis is basically morphogens that are specific to a certain feature are more concentrated in that end of the unfertilized egg. So basically this is the cytoplasmic determinants that will decide what types of cells the embryo will have. So here in the unfertilized egg, there will be a lot of bicoid mRNA concentrated here. And then this will lead to more bicoid protein on the same end in the early embryo. Okay, so here's some genes that are associated with cancer. So oncogenes cause cancer, and proto-oncogenes are coding for normal proteins. So these proto-oncogenes can become oncogenes through a lot of processes. So this includes translocation or transposition, where the proto-oncogene will get moved to another location under new control that can be a lot more active than the old one. So also the genes can be more amplified with more copies of the gene that results in more total transcription and then more protein formed. So lastly, they can also become oncogenes by point mutations in the control of the gene or the gene itself. Okay, so as opposed to oncogenes, tumor suppressor genes will actively inhibit cell division. So these genes will produce products that can repair DNA or control the adhesion of cells to each other or attachment surfaces, and this is lacking in cancer cells. So other proteins can also help inhibit the cell cycle of cancerous cells. These genes are very important, and mutations that decrease the activities of these genes can contribute to cancer, since the tumor growth, the uncontrollable cell growth cannot be controlled anymore. Okay, so here's some examples of interference with normal cell signaling pathways because of genes. So the RAS gene, which is named for rat sarcoma, in which it was first discovered, it encodes for a G protein that relays signals from tyrosine-dependent kinases. So under normal conditions, the RAS protein isn't active, and it's only going to be activated if there's a signal. However, if there's a mutation in the RAS pathway, the G protein will be constantly active even though there's no signal. So then this will result in excessive cell growth and division that can cause the cell to form a tumor and potentially become cancerous. So additionally, the p53 gene is a really well-known tumor suppressor gene. So this gene will normally activate microRNAs that will stop cells that have DNA damage during the cell cycle for DNA repair or apoptosis if the DNA damage is too severe. But if mutations in the p53 gene occur, that will result in division of cells with damaged DNA that can cause the cell to be cancerous. So also, there are certain viruses that can cause cancer, such as the papillomavirus. So the integration of viral genes can donate oncogenes to cells, which can then be incorporated and cause cancer in cells. Okay. Let me see if there's questions. Oh. Okay, moving on. Okay, so speaking of viruses, we're going to talk about viruses. Okay, so chapter 19 is viruses. So viruses contain either RNA or DNA that is enclosed in a capsid or a protein shell. So the virus will be basically named based on its genetic material, if it's RNA or DNA. So the capsid, or like the outer shell of the virus, can have many shapes. And the most common are rod-shaped, polyhedral, enveloped, or complex, like a bacteriophage on the bottom here. These capsids are mostly composed of capsomeres, but there can be also other proteins that are integrated into the capsid. So viruses can also have viral envelopes to help with infection, and those envelopes are usually derived from the host cell's membrane, which contains cell-specific glycoproteins and surface proteins that can help the virus disguise itself and then make its way into the cell. So this happens since the viruses will break out of the cell's membrane and will take a little bit of the membrane each time with itself. So a special type of virus is called the bacteriophage, which has the complex shape here. So these bacteriophages will infect bacteria, and they're really useful in a lot of studies and experiments. And yeah. So this is how bacterial viruses will replicate. So this is the lytic cycle. So the lytic cycle for bacterial viruses is caused by the virulent phage, which ends in the death of the host cell. So first, the bacteriophage will attach to the bacteria with surface receptors, and then it will pump its own genetic material into the bacteria, and then this genetic material can cause the bacteria's DNA to become cut up. So then with the viral genetic material, the bacteria cells will make the viral genomes and proteins in the viral parts, which will then self-assemble and then burst out of the cell. So this will result in a very violent death of the host cell as it bursts open with new viruses. And as you can see, the DNA of the genetic material of the bacteriophage is immediately used up and formed into viruses. So the next type of cycle is the lysogenic cycle. So this involves replication of a phage genome without destroying the host immediately. So this phage genome will actually be incorporated into the host cell's genome without being transcribed immediately. So these phages are called temperate phages, and this genome will be incorporated into the cellular DNA and then passed on during mitosis into other daughter cells. And so that is why these viruses can be transmitted so quickly. So after the lysogenic cycle, so after a couple of rounds, the cycle can actually turn lytic, where the bacteria will then break out, but it depends on every virus's timing that they utilize, I guess. Okay, so to defend against viral genomes being replicating or being incorporated, bacteria have certain restriction enzymes that can help cut up the viral genome. So bacteria can have a CRISPR-Cas9 system, which will basically put DNA of the viruses in between two repeat or palindromic sequences. So the next time the same DNA is put into the cell, the Cas9 will bind to the viral DNA with the help of a guide RNA to help cut the specific section of viral DNA, and then basically eliminate the threat of the viral genome. So animal virus replication is a little bit different because the animal viruses don't look like little space alien things. So they will use the viral envelopes to enter the host cell. These envelopes contain glycoproteins that will bind to receptors on the host cell surface. So it helps them, it's basically a disguise for them to enter the cell. So the cell's machinery will assemble the viral parts with the viral genome, and then transport it to the cell surface. So when the cell emerges, it will wrap itself in another layer of the host cell's membranes to disguise itself and infect the next host cell. So retroviruses will have reverse transcriptase because it contains RNA initially. So reverse transcriptases will transcribe the RNA into DNA into the host cells. So an example of a retrovirus is HIV, which causes AIDS. So their genetic material is called a provirus that never leaves the host's genome. So to help defend against virus infections, or deadly virus infections, vaccines are basically weakened or dead or viral parts that are injected into the body or taken orally, so that will help the body build up its defenses for viral infections. So just some terms though. An epidemic is a widespread outbreak. An epidemic is a global epidemic, so that's what we went through a couple of years ago. Something similar to viruses though are prions. So prions are very interesting because they're misfolded proteins that will cause degenerative brain disease in a lot of animals. So prions will actually lead to mad cow disease that can spread from cows to humans when we eat the meat. So prions can convert normal folding proteins to being misfolded, which leads to very widespread transmission and infection, and there's no cure as of now. So, for example, if you eat a cow with mad cow disease, you'll get the prions as well, and then you'll start to have the degenerative brain diseases that the cows originally had. So I don't know if you have heard of amyotrophic lateral sclerosis, or ALS. So ALS is also neurodegenerative, and part of its causes, so there are many causes, but part of them could be so there is studies going on with that, but it is pretty much proven that one of the causes is protein misfolding buildup. So these are not technically prions, but they're very similar to prions in that the misfoldings and aggregations can so these misfoldings and aggregations can build up and basically help spread ALS. Oh, Anthony, I'll go a little bit into 20, but you can finish the rest if I can't finish. Okay, so chapter 20 is the last chapter. I'll probably go a little bit into it, and then Anthony will finish up next time. Okay, so Camel 20 is DNA tools and biotech. Cool. Okay, so first is DNA sequencing, where you try to figure out what nucleotides the DNA has. So the DNA will be cut into fragments, could be cut into fragments and sequenced. So the first procedure to do so is dideoxyribonucleotide chain termination sequencing. So basically, there will be fluorescent dideoxyribonucleotides and also normal nucleotides that will basically synthesize the chain. And these dideoxyribonucleotides, they lack another oxygen, so there will be no more nucleotides that can be added on after that. And so these chains will be of different sizes. Those will be run through gel electrophoresis and then sequenced through laser excitation. So the next procedure is sequencing by synthesis. So basically a specific strand of the fragment will be immobilized. Complementary strand will be synthesized one nucleotide at a time, and that will be sequenced. And the third major procedure is nanopore sequencing. So the single DNA strand will be moved through a very small pore and sequenced through an electrical current disruption analysis. So I'm going pretty fast, but these are pretty detailed procedures. So first, this is dideoxyribonucleotide sequencing. So there are fluorescently labeled dideoxyribonucleotides. So these will basically be like the end nucleotide of every chain, and those will be synthesized. Yeah, okay. And those will be sequenced through laser beam excitation into a graph that looks like this, and then you can basically tell, oh, which nucleotide is it right now. They will also have like different colors. Okay, this is what's it called again? Oh, this is sequencing by synthesis. So basically, there is a complementary strand, and then the other strand is sequenced one nucleotide at a time. And then this is nanopore DNA sequencing, where the DNA will be unwound and then put through a nanopore where there's an ion current that analyzes what nucleotide you have there. Okay. Next is DNA cloning. So scientists can basically insert plasmids with external DNA that they want, or it's called recombinant DNA, into bacteria. So the bacteria can replicate with the recombinant DNA since the plasmid is like a part of their genome now. And this is called gene cloning. And the plasmid is called the cloning vector. So this allows for amplification of a particular gene and its protein product, which can help with mass production of a product. So this is since bacteria can replicate super quickly, so they will have a lot of the protein product and the gene that gets inserted. Okay, so here's how recombinant plasmids are made. So restriction enzymes, which I mentioned before, they will cut up DNA. So these restriction enzymes will cut restriction sites on the DNA to allow for insertion of new sequences. So DNA is usually cleaved in a staggered manner. So these are called sticky ends that can kind of stick back together with the help of DNA repair mechanisms. So they are rejoined by DNA ligase, which we have seen in DNA replication before. So restriction enzymes are very specific to the site that they can cut on DNA. So yeah, that's important to know. And these can be separated by gel electrophoresis, which separates DNA fragments based on size, and then this can be used to analyze restriction sites. So in gel electrophoresis, there's and a cathode that have a positive for the DNA to go through. So DNA is negatively charged because of its phosphate groups. So it will tend to move towards the positive charge. And since bigger DNA will be trapped more by the gel, so the smaller fragments will move faster down the gel. And that's how you tell how big your segments are. Okay, so next I'm DNA using PCR. So PCR stands for polymerase chain reaction. This allows amplification of the desired gene. So there are 3 steps. The first step is heating, second step is cooling, and then the third step is heating again. So first, the double-stranded DNA has to be denatured by heat. So all of the hydrogen bonds in the middle will be basically broken by the heat. Sorry. Step 2 is cooling to allow the attachment of DNA primers so that the polymerase can attach the DNA. Step 3 is heating it up again to allow heat-stable polymerases to extend the primers. So these polymerases have to be very heat-stable because the third step is very hot. And so these polymerases are often Taq polymerases, which come from thermophilic bacteria or bacteria that always live in heat, so the polymerase will not denature. Okay, so next is expressing cloned eukaryotic genes. So in bacteria, an expression vector will contain a very highly active promoter upstream of the restriction site. So this will promote expression of the foreign gene that's inserted into the restriction site. And also complementary DNA, which is from RNA, and only including the exons can also be used. In eukaryotes, the DNA can be put into the cell through electroporation, which is basically when an electron pulse is used to basically make tiny holes in the cell membrane, and then that can allow the DNA to go into the cell. Okay, it's 8, so I'm gonna finish at 20.1, so Anthony can continue with 20.2 next time.